I would like to thank the AMMF for the invitation to give a talk. I would like to thank the chairs for the introduction. So the reason I changed the title because I update a bit the other sort of data, but see how much time I have. Basically, this is very preliminary and ongoing studies, and open to your comments and seek your support as well as collaboration. So just because of science, we just kind of start with very sort of brief about the tumor initiation and the progression. So the process is multi-step, evolutionary process. So it begins with the somatic mutations and then eventually progress to the malignant formation. So in the normal tissues, somatic mutations occurred sporadically. Fortunately, not all of them progress to cancer. And this is maybe either tumors were eliminated by tumor suppressive mechanisms, or they remain kept in check by normal tissue architectures and the homeostasis. So the real shift occurs from the stage of normal to malignancy is when the mutated cells exposed to environmental insults. So Professor Banalis mentioned quite a lot about the risk factors in the last sections. I'm not going to elaborate it. So these changes will make mutated cells acquire more additional mutations, and therefore they remodel the tumor microenvironment by driving the stromal cells recruitment, activation, and differentiation. So over time, Darwinian selection by environmental remodeling favors the fit clones to divide and driving tumor progression. So the concept of tumor microenvironment is modern revival of the seed and soil theory, which was first published by Stephen Pages, Oxford graduated surgeon in 1889. The real tumor microenvironment gained the scientific momentum during the 1980s and 1990s, when we beginning to understand the tumors. Rather than viewing them as a collection of the mutated cells, we recognize them as kind of complex and abnormal organs made up of the tumor cells and the surrounding stromal cells. We understand that tumor microenvironment is consisted of the cellular, non-cellular, and the cellular compartments. The non-cellular sides, including the extracellular matrix, growth factors, cytokine, chemokines, metabolites, and other many soluble factors. On the cellular sides, it includes very famous cancer-associated fibroblasts because it consists of roughly about 70 to 80% of the solid tumor structure, in addition of the endothelial cells, parasites, and even more hot topic about the immune and the inflammatory cells. because it involved in the immune evasion. We understand the tumor microenvironment as playing a critical role in the promoting tumor growth. However, the epithelial cells, including the cholangiocytes, their role in the tumor microenvironment was not as kind of fully incorporated in what we think about the tumor microenvironment. They could participating in modulate the tumor microenvironments and influence the cholangiocarcinoma cells growth. Cholangiocytes, as well, I don't need to talk too much, just briefly take it over here. So cholangiocytes line and interconnected the intrahepatic and the extrahepatic bile ducts. They modulate the bile conditions by regulating the transporters for the and control the ions, solutes, and water in the bile duct lumens. The cholangiocytes are functionally heterogeneous, as well reviewed by Professor Banalis in 2019, and recently others. But overall, in the healthy livers, so cholangiocytes are generally quite quiescent. But in response to the injury or stress, they become active and participating in the liver repair. Under so the chronic disease states, they could act as like a pro-inflammatory here and pro-fibrogenic, even pro-tumorigenic functions. However, the role of the cholangiocytes in the regulation of the cholangiocarcinoma growth is not well defined. We just wanted to ask a question: how do cholangiocytes influence cholangiocarcinoma growth, particularly during the early onset of tumor development? To address this, we set up a co-culture model whereby growing the cholangiocyte, which is MMNK1, and the human primary cholangiocyte, which is NHC3, kindly shared by Professor Banalis. And the cholangiocarcinoma cell lines, we have 3 intrahepatic cholangiocarcinomas, including SW1LP11196, and the extrahepatic cholangiocarcinoma, so which is SNU1079, and kindly shared by Professor Stuart Forbes at University of Edinburgh. So we grow them in a harsh, less permissive culture conditions, which is with low glucose and 0.1% BSA instead of the feeble bovine albumin for 40 hours. This setting, we try to mimic the harsh tumor microenvironment and trying to catch the features of the interactions in terms of the omics features at early stages. So then by leveraging of the multi-omics technique, we try to study the interactive dimensions of the transcriptional regulation, cytokine secretion, and the cellular metabolisms. So in this case, I think today possibly I'm just going to walk you through a bit more on the metabolism. And if I have time, I'm going to recap about the transcriptomic response and the cytokine profiling during the early interactions. So firstly, let's see what effects on the functional effects on the co-culture model. So you can see it's quite busy slides, but let's focus on, to start with, to focus on this model. So using FACS analysis, we found the co-culture MMNK cholangiocytes decrease the tumor cells viability and increase the tumor cells apoptosis, while the effect on cholangiocytes itself was not significant. So we validated this finding across in 3 other different cholangiocarcinoma cell lines, including OP1, SNU79, 1196, and so on. So the pattern is very similar. Furthermore, co-culture cholangiocyte also prevented the cholangiocarcinoma cells transcell invasion compared to monoculture, which is the same case as in the LP1 compared to the co-culture. And immunoblotting confirmed the co-culture downregulated a mesenchymal marker vimentin. Vimentin is an important marker playing a critical role in the tumor migration. So in summary, this functional phenotype during the early 48-hour co-culture, non-cancerous cholangiocytes restrained the cholangiocarcinoma cell growth by decreasing the tumor cells viability, induced the tumor cells apoptosis, and suppressed the tumor cells EMT-mediated invasion. Now, talking about a bit more heavy about the science here and technique, but I want to dive in for details. And here is the omics. So omics is currently is very advanced technologies for us, helping us to look at different levels change of the features of the gene, protein, epigenetic alterations, and metabolism, and so on. So let's focus on the metabolism here. So it is well known that the tumor cells reprogramming the surrounding microenvironment, including itself, which is a hallmarker of cancer. So in contrast to the normal cells, tumor cells, cancer cells, reprogram the metabolism by increasing the glycolysis and lactate production, even in the presence of oxygens, while maintaining mitochondrial activities, which make tumor more adaptable to the hypoxic conditions, and they make energy turn over quickly and increase the biosynthetic pathways for the rapid growth and the invasion. So then, to investigate the metabolic role in the regulation of the cholangiocyte and cholangiocarcinoma interaction at early stages, so we use the co-culture. Then we prepare the cells, harvest cell samples at 3 different time points. This helps us to catch the metabolic shift at different time points. So we observed that the distinct co-cultures of the cholangiocytes and the cholangiocarcinoma cells metabolic file completely different. So compared to the co-culture, compared to monocultures, we found that there's an increase of the choline and creatine production in the cholangiocyte co-culture. Because the creatine production can be derived from the choline through a glycine intermediate. So then we ask the question, whether creatine can play any impact roles in the cholangiocytes cell viability. Then we supplemented cholangiocytes with the creatine and measure the cell viability, and we found indeed the creatine increased cell viability in a dose-dependent manner. So this makes us to think about where we suggest that maybe cholangiocytes interact with the tumor cells itself, then increase choline uptake, and then convert to creatine, then increase ATP buffering and maintain the energy balance, and therefore to compete with the tumor cells for survival. Now, the profiling of the metabolism in the, you can see it, cholangiocarcinoma is very different, as mentioned. So the whole profiling, you can see a lot of them are red, a lot of them are blue. So just give you an idea, the signature are very different of the co-culture, completely different from the monocultures. Then we observed, interestingly, one metabolite, which is 2-oxoisopentanoate. So to start with, we tried to find out exactly what it is. We went through so many textbooks, it's unknown. Then one day I had to contact the head of department of chemistry at Oxford University. Then he told me what it is. And so it's rare, and it's completely unknown, along with a few metabolites, glycolytic metabolites. So now here is a box plot, show you the efforts of the 2-oxoisopentanoate production in co-culture compared to monoculture. So nothing over there. So therefore, the question would be, what is it 2-oxoisopentanoate? Or I'm going to make short like oxip. So in co-culture, we see the accumulation of the oxip in the cholangiocarcinoma cells. It suggests that the co-culture modulated tumor cells branched chain amino acid metabolism. So the oxip is a metabolite of the branched chain amino acid. So branched chain amino acid include 3 essential amino acids, which is leucine, isoleucine, and valine. And they are taken up into the cells through the L-type amino acid transporters. And they can be catabolized to make the DNA, RNA, and the protein in the cytoplasm. They also can be further uptaken by the mitochondria through this transporter, then enter the TCA through a few more steps, enter TCA cycles for energy production. As I said, the role of the isopentanoate or oxip in co-culture is what we show here is at 48 time point. But here I try to give you details about the competition between normal cholangiocyte and cholangiocarcinomas, kinetic shift for in 3 time points. You can see in the blue where the branched chain amino acid uptaken by the cholangiocytes in a kind of upward trends fashion. In contrast, the branched chain amino acid uptaken in the cholangiocyte carcinoma cells co-culture pretty much stayed, or a later time point dropped a bit, and maybe that is a kind of the downward trend, maybe. But so as said here, so therefore we found basically OXIP, or another two names like KIF, or another one was a methyl 2-oxybutanoate. So they are just the same thing here. Okay, so in run out of time. So this is the production of the pathways, and appear to be normal cholangiocyte compete with the cholangiocarcinoma cells for energy production, protein synthesis as such. Then we try to evaluate the functional role of the OXIP in the metabolism. So we using the FACS analysis, then we treated the cells, cholangiocarcinoma cells, with the OXIP, and we found there was an inhibition of the cell viability and increased apoptosis in the cholangiocarcinoma cells. Following the validation in additional 3 cell lines, the pattern is very similar. Then we also conducted the immunoblotting. We found there was increased the apoptotic markers, which is caspase-3. So furthermore, treatment of the cholangiocarcinoma cells with the OXIP, you can see there was a dose response inhibition of the tumor cells transfer invasion, and this corresponding with downregulation of the mesenchymal marker vimentin. Importantly, we found that actually when there was a prominent of the inhibition of the cholangiocarcinoma cells clonogenesis by the drug. By the inhibitor, it's no drug yet. So the effect on the normal cholangiocytes was not significant, and there was quite dramatic changes of the colony formation in the SW1079, be more extreme, but still slightly changes. I mean, inhibition of the formation in other 2 cholangiocarcinoma cell lines. So in summary, we found that basically the co-culture and cholangiocytes modulate the metabolic environment, and the role of this metabolites appear to be inhibitory on the cholangiocarcinoma cells growth. Currently, we're trying to evaluate it, OXIP, in the cholangiocarcinoma organoids in collaboration with group in the Keio University in Japan. And hopefully, I talked to Laura the other day, we could evaluate it in your patient-derived tissue plants for the study. We're also trying to formulate the compound into nanoparticles, which is currently ongoing, and hopefully we could also test it in like animal models, and open to collaboration if you are available. And so this transcription profiling, I think I have run out of my time, so I just skip to the conclusion, and hopefully I will present more about this in a complete study in the future. So this overall, these findings just show multi-layers of the cholangiocyte and cholangiocarcinoma interactions, while the potential, the OXIP, maybe so, and its related pathways maybe a therapeutic targets in our models, but I don't know how effective it would be in other models. So we will continue to investigate it. Otherwise, thank you very much for your attention.